I would like to ask a very dummy question with the non-specialists, and there are many out there, and certainly I'm one of them. I'm simple-minded heart failure doctor. You speak very frequently about Mendelian randomization. What is it this altogether? How this is helping you in your area of lipid clinical trial, Bob?
Yes, so Mendelian randomization is identifying a certain trait that regulates a lipoprotein pathway and then evaluates whether this pathway, typically loss of function variants, sometimes gain of function variants, are associated with ischemic heart disease. And if you identify that genetic association, you feel more confident that that's a good target. And there's been this explosion in new therapies that target the message for some of these proteins that regulate these diverse pathways, be it angiopoietin-like 3, ApoC3, lipoprotein A, PCSK9 is another example. Certainly we have to do the clinical trials, but it gives us a little more comfort that we're going in the right direction with our pharmaceutical development.
That's really smart, and certainly you have multiple ways in your area of enriching the patient population because you can phenotype the patient on multiple ways, Mendelian randomization, genetic, and the different fraction of lipids, etc. But then you also mentioned the inflammation and the anti-inflammatory atherosclerotic medication. If you are with a drug acting mainly as an anti-inflammatory drug, where you should start? Would you select patient on the basis of CRP or some other inflammatory markers? And the second question I'd like to ask you so far is, industry are usually wary about having a companion diagnostic and therefore having an indication whereby above and beyond the lipid level, they would need to screen their patient for CRP for whatever reason. So what is your take on this, Wolfgang, maybe?
Yeah. I think you're perfectly right. What I wanted to say in this context, if you have if you look into anti-inflammatory treatment, certainly there must be some inflammation in the background. Otherwise, you wouldn't do that. Now, there's some recent large-scale analysis by Paul Ridker based on three huge studies, clearly showing that in CAD patients, despite adequate lowering of LDL, there is a considerable pro-inflammatory risk that needs to be tackled to further improve the situation in these patients. So you can either go by phenotype. I mean, ACS patients certainly, in many instances, do show some signals of pro-inflammatory response. You may use, as we have done in Jupiter, and also in particular in Cantos, a proof of concept study for the anti-inflammatory therapy, to use the CRP as a marker. There may be others. I mean, we have recently done a secondary prevention study with proteomics, where we also looked into potential inflammation in those with a CRP below 2 milligrams per liter. And indeed, there were a number of other pro-inflammatory markers involved. So there's certainly something out there beyond the C-reactive protein. What we are mainly interested right now, and clinical research goes in this direction, since we have identified based on the canakinumab and L1 beta inhibitor, and in the context of the proof of concept study Cantos, that the NLRP3 inflammasome may play a critical role. A lot of energy goes into identifying other players in that context. For example, IL-6 antibodies. There's clinical trial underway right now, the Zeus trial and the Hermes trial and HFpEF, as you know. There are other ways that are being followed by certain companies looking into specific inhibitors of the inflammasome. So this is a little bit of a focus. Whether this is the story by itself, we don't know, but the NLRP3 inflammasome looks very promising to be followed further on and to look for further candidates that might be safe and effective to reduce the residual inflammatory risk.
Yes, Bob, we have discussed about the multiple biotargets, and certainly a better way to define the patient population and enrich risk, selecting patient with specific biotargets, that's lipid fraction or inflammasome indicators, and these are what we call liquid biomarkers. But now there are growing interest in imaging. Patient with imaging of atherosclerosis, which may be markers of risk, whether this is helpful into identifying patients most likely to be responders or most likely to have a high event rate. What is the state of research on imaging in atherosclerosis in defining the target patient population?
Well, Faiez, I think imaging is of great potential. And we know that certain characteristics of the coronary plaque, the lipid-rich plaque, or the vulnerable plaque index, is an important predictor of future cardiovascular events. So it's imaging tied to events using those metrics. And when we think about more the primary prevention population, not everybody who's got an abnormal biomarker will have a cardiovascular event in their lifetime. So in order to enrich a more primary prevention or subclinical atherosclerosis study, it would be worthwhile knowing whether they actually have plaque present, typically in the coronary arteries. It's looking at imaging to help with the appropriate recruitment is very important. I'm quite intrigued that we have patients who have very high levels of lipoprotein A, but who have no disease by coronary CT angiography. And these are individuals that, based on their other risk characteristics, we would expect to have disease, but something's protecting them. There's something at the level of the vessel wall that is mitigating against these atherogenic lipoproteins. And so in order to move forward, let's say with primary prevention, or even individuals with intermediate risk based on their pooled risk equation or other equivalent techniques, determining whether they have disease already, I think is an important path forward. So we don't end up giving therapies to people that are just never going to develop, disease. Of course, the clinical trials right now are focusing on the extremely high-risk individuals. We have trials with, LPA-lowering therapies and inclisiran that are looking at very high-risk individuals, those who have had an MI, had a stroke, have symptomatic PAD. But the question is, can we look at a population that's much broader, ones that are still at high risk, but not at that extremely high risk category?
So, if I may just
absolutely
one or two sentences, Bob is perfectly right. There is a huge potential with regard to imaging methods to get involved in clinical trials, and the beauty really is that we're not dealing with floating biomarkers in the circulation, but we look into the vessel wall where the atherosclerosis takes place. Now, to my knowledge, so far, CTA has developed a number of, and Bob has already mentioned some of them, very nice criteria that have also been validated. Maybe not so much in CTA, but rather with intravascular ultrasound, with near-infrared spectroscopy, etc. But what has been done so far is looking into those biomarkers from prospectively whether or not they are really indicators of a vulnerable plaque, and it seems to be. But what I have seen in terms of clinical trials, this is still a missing part. We have not included. I wouldn't know of any trial that has included specifically patients, let's say, with a low attenuation plaque or some kind of vulnerable plaque criteria as a marker of very high risk. So that probably is an area that needs to be followed in the future.